Hello, everyone, and welcome to today's webinar, Quality Control Electronic Records for 21 CFR Part 11 Data Integrity. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit beckman.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now present today's speaker, Tony Harrison, Senior Marketing Manager at Beckman Coulter Life Sciences. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Tony, you may now begin your presentation. Welcome.
Thank you for that introduction. Hello and welcome everyone to this short seminar today on quality control, compendial testing, and 21 CFR Part 11 data integrity. My name is Tony Harrison and I'm Senior Manager for Beckman Coulter. So today's presentation, as I say, is quite short. It'll last approximately 30 minutes, and then of course, plus the question and answer session at the end. Now, this talk takes a look at 3 compendial quality control test procedures and how automation of those procedures can help improve data integrity by reducing the opportunities for human error. The 3 quality control procedures discussed in this presentation form a common thread in the biopharmaceutical manufacturing industry and are water quality testing, clean room routine environmental monitoring, and final injectable drug particulate testing. So first, let's take a look at quality control testing and how this can be open to human error, unfortunately resulting in data integrity issues. Now, in 2018, December of 2018, the FDA published their questions and answers paper relating to 21 CFR Part 11 data integrity. And they shared some fairly worrying observations. They stated that in recent years, they'd increasingly observed cGMP violations, particularly involving data integrity during their inspections. And they said that these violations have led to numerous regulatory actions, including warning letters, import alerts, and consent decrees. And they go on to say that firms should implement meaningful and effective strategies to manage their data integrity risks. And what they're suggesting is that you should make processes more robust. Also in their 2018 guidance, the FDA introduced the acronym for the first time, ALCOA. And this is in response to what a good electronic record should look like. And they say the acronym stands for attributable to the person taking the sample. They suggest it should be legible and therefore probably not handwritten. Sometimes handwriting can be difficult to read. They say it should be made contemporaneously, which means at the time of the test. So the electronic record should be made at the time of the test and rather than later. And they're referring there really to the common practice of taking paper printouts from test machines and then manually typing that information in to create the electronic record at a later time. And they're saying that's not good practice. And they also say, of course, the electronic record should be the original one and the data it contains should be accurate. And it's that last accurate, A for accurate, that I'm going to focus on today. Now, in a typical manual sampling and data quality control situation, there's many manual steps in the SOP. And first of all, of course, the technician has to make sure they're using the correct version of the SOP. And then they have to manually configure the instrument to sample for the correct volume and the correct number of samples according to the SOP for each different test. Next, they print the sample results out and handwrite the comments onto the printout and sign it with their name to make it attributable. And then, at the end of each day, they manually type the results in to create the electronic record from all these pieces of paper they've created. And finally, the electronic record has to be reviewed by the supervisor, A, to ensure that all the samples have been taken correctly, and B, that the data in the electronic record matches the paper printouts. Now, as you can see, this is a very manual process, and with thousands of samples, pieces of paper, and manual data transcriptions every month, the opportunity for human error to creep in and impact the integrity of the data or the accuracy of the data is very high indeed. So I wanted to share with you a study which was done by the Industrial Biodevelopment Laboratory, and they showed in a test between 16 experienced QC technicians performing the same manual SOP standard operating procedure, a variability of up to 20% between the results. Now, you could argue that the manual SOP could be improved and the technicians retrained, but such variability in results does suggest that manual processes are arguably not robust enough to deliver the required levels of quality control. Now, whilst the technical specifications of similar instrumentation may appear equivalent, when looking to improve quality control, the QC team should really be looking at the level of automation that the instrument offers to make QC testing more robust and repeatable. For example, a common source of errors in QC testing is sample preparation. So any instrument that automates this process can enhance the reproducibility and the robustness of a QC test. So let's take a look at our first quality control test, the quality control testing for purified water and water for injection. And in fact, in this talk here, I'll talk about how we can use online instrumentation that automate this testing and use that data from them to help release batch files. Now, total organic carbon, or TOC, and conductivity are two of the four critical water quality attributes defined for water for injection and purified water in the United States Pharmacopoeia. Now, online analyzers, such as the PAT700 from Beckman Coulter, can be validated to comply fully with both of these key quality attributes according to the Pharmacopoeia requirements. The Pharmacopoeias ask that TOC is measured by first measuring the inorganic material present in the sample, total inorganic carbon they call this, or TIC, and the Pharmacopoeias guide us that TIC is mainly dissolved carbon dioxide in purified water and water for injection. The Pharmacopoeias then ask that the organic material present in the sample is now completely oxidized to release the organic carbon in the form of carbon dioxide. The carbon dioxide from the oxidized organic material and the inorganic carbon dioxide now add together to represent the total carbon present in the water, or TC. And then the Pharmacopoeias ask that we subtract the TIC from the TC to calculate the total organic carbon, or TOC. Now, this is a graphical representation of the oxidization process. Initially, the conductivity of the water sample is measured to establish the amount of TIC present in the sample, which, as I said, is made up of carbon dioxide dissolved in the water. Then an ultraviolet lamp is used to oxidize all the organic material in the sample to carbon dioxide. And when there's no further changes in the conductivity, we know we've completed that oxidation, and the analyzer measures the new conductivity value, which is the total carbon, or TC. And then it subtracts the initial conductivity measurement, the TIC, from the TC, and the difference in this is directly proportional to the TOC in the water sample itself. Now, traditionally, data from an online TOC analyzer is fed straight to the site's supervisory control or data acquisition system or distributed control systems, SCADA or DCS, and making access to this data for QC team difficult. However, the PAT700 can be configured to automatically send PDF electronic records every day over the Ethernet for review and batch release. And this satisfies the FDA's ALCOA requirement for electronic records to be accurate, legible, contemporaneously created, original, and attributable. Now, unlike laboratory water quality testing instruments, which utilize reagents and require manual configuration for each sample and frequent, sometimes daily calibration, the online PAT700 from Beckman Coulter requires no reagents. It automates the test protocol and only requires calibration every 12 months. Calibration and system suitability testing SOPs are automated, requiring no manual data entry, and pass-fail reports are automatically generated from the instrument, eliminating manual calculations. Calibration standards, once loaded into the PAT700, upload their certified value, lot number, and expiry date to the PAT700 using radio frequency identification. Electronic chips are located on the calibration bottles, and these are read by the PAT700, thus avoiding manual data entry. Okay, so let's move on to look what can be done to automate routine clean room environmental monitoring programs. Now, this of course falls under the GMPs, and there are two main flavors of the GMPs. This is the American, North American cGMP, and the European and World Health GMP Annex 1, which is the same document. So everywhere in the world follows the European Annex 1, apart from North America with the cGMP document. Now, both these GMP documents, however, call up the ISO standard ISO 14644 Part 1 for the methodology of clean room classification. So let's take a look at the GMP regulations controlling the contamination levels in these clean rooms. Now, this is the current table for the air particle contamination levels in the European GMP Annex 1. And as I said, it's the same in the Guide for Sterile Manufacturing from the World Health Organization. Now, I say the current table because currently the Annex 1 is under review. However, that's been happening over a period of many years, and the last version of the Annex, proposed new Annex I saw, was version 12. So I'm not holding my breath for the next one, but it will get revised. And you can see there's 4 grades here of clean room, from the very cleanest grade A to the dirtiest grade D. And you can see that the European GMP calls for 2 particle sizes to be measured, both 0.5 and 5 microns. Now, this is the same the world over, because the World Health Organization follows the same. And the only difference is the North American cGMP, which only calls for 0.5 micron particles to be measured and reported. All particle counts are reported in particles per cubic meter for both GMPs, and this itself can present an area of potential error, an area of potential error, because in order to reduce the amount of time taking typically in routine environmental monitoring programs, the sample taken at each location is often just 1 minute sample. Now, with a 1 cubic foot per minute particle counter, that means that the sample volume taken is just 1 cubic foot of air, and now there are approximately 35.2 cubic feet of air in 1 cubic meter. So users must multiply the counts results by 35.2 to get the counts per meter. So you can see the opportunity for error there. Now, it is true that most particle counters can be programmed to automatically make this calculation for the user, but it's another configuration step to remember to do to ensure the results are reported correctly. And I have seen people not doing that and just writing down the counts per cubic foot. Now, as I mentioned, the North American FDA cGMP document does differ to the European and World Health Organization guidance in that it only mentions one particle size, the 0.5 micron particles. However, of course, drug manufacturers based in North America wanting to sell their products outside of North America will have to count both 0.5 and 5 microns, as they have to be in compliance with the standards of the industry and the country to which they want to export. Now, as I mentioned a couple of moments ago, both the European GMP and the North American GMP tell the reader to refer to the classification procedures laid down in the ISO standard 14644 Part 1 for classification. Now, this in itself is not a GMP document and is designed to give guidance rather than mandate, but the fact that it is referenced by the GMPs gives it the same status as a GMP document for those manufacturers regulated by GMP. Now, it has a wide range of cleanroom classes, as you can see, from 1 to 9, and it covers, again, a wide range of particle sizes from 0.5 microns up to 5 microns. And then it defines in the table the number of particles for each class. You'll notice this is the 2015 version of the ISO standard here. And for those of you with sharp eyes will notice that at ISO class 5, for 5 microns, there's no number in the table anymore. That was removed in the 2015 version. However, if you're complying to the European GMP or World Health Organization, that 5 micron particles is still in those regulations. So even though it's gone from the 14644 document, you still have to measure those if you're complying with European or World Health Organization rules. So the idea with the ISO standard is the user must decide what particle sizes they're interested in and how clean the clean room must be. And then they choose a subset of the clean room classes and particle size from the ISO table. Now, GMP, the European version, has decided that particle sizes of interest are 0.5 and 5 microns. And you can see from grade B at rest, if we go to the grade B table here, 0.5 and 5, and the number of particles in the table there are the same as ISO class 5. So you can see how GMP has taken a subset of the ISO to make the GMP regulations. And don't forget, if you're following European GMP or you are following World Health Organization, it is 0.5 and 5 microns still. Now, discussions that I've had as I travel the world and speak to users of clean rooms in the GMP environment is that there's an increasing trend where the burden of carrying out these routine environmental monitoring testing, particularly air particle counting, is moving away from the QC microbiology team and over to the production staff for two reasons. Firstly, the microbiology staff are very overqualified, shall we say, to carry out such simple routine tasks. And the second point is it reduces the number of people in the clean rooms, thus hopefully reducing the potential for product contamination. However, the downside is the production team do not, of course, have the same high level of knowledge about microbial and routine environmental monitoring, and this creates challenges in itself. Now, routine environmental monitoring is arguably one of the most manual and complex processes that we have to do. It involves the collection of thousands of samples each month using portable air particle counters. And typically there are many manual SOP steps to follow in the routine environmental monitoring program. First, of course, the technician has to make sure they're using the correct version of the paper SOP. And then they have to follow the sampling map in the SOP and go to every sample location. Then they have to manually type the location name into the counter via the on-screen keyboard. And next they have to configure the counter to sample for the correct amount of time, the correct volume, and the number of samples according to their own SOP for each location. Next, they print the sample results out from the counter and handwrite any comments onto the printout and then sign it with their name. And at the end of every day, they photocopy all these printouts and then they manually transcribe the results from every piece of paper to create an electronic record. Then finally, at the end of the day, the electronic record is reviewed by the supervisor to try and ensure that all the samples have been taken correctly and that the data in the electronic record actually matches that of the paper printouts. So as you can see, this is a horrible manual process, and with thousands of samples, pieces of paper, manual data transcriptions every month, the opportunity for human error to creep in and impact the integrity of the data is very high. So with the new MET1 3400 plus particle counter from Beckman Coulter, we've tried to address these issues with the Clarity software which runs inside the counter itself. First of all, it improves the accuracy because it allows you to upload your own sampling map from your routine environmental monitoring SOP into the counter, creating an interactive version of that sampling map on the screen of the counter itself. Now, Clarity software includes, as I said, the interactive sampling map on the counter screen. The electronic records are created contemporaneously and can be exported via built-in Wi-Fi or wired Ethernet, and this is standard. It exports these in secure PDF, they're password-protected PDF, or you can get it in Excel if you want to trend them. And there's a tab-separated variable format for LIMS integration. It uses Microsoft Active Directory for username and password control, so it's very secure. And the data exporting function is LDAP, which is the currently regarded as the most secure way of doing this. This time, SOP version control is done in the counter itself, and the name of the SOP that's been used on the day, plus the version number, appears inside the electronic record, so you have high traceability. Now, the counter can work with a USB barcode scanner, and it can scan the microbial plates used at each location, so they then appear in the electronic record as well. So that way you're tying the settle plate and the active air plates that you use at each location in the clean room to a date and a time. So this gives traceability for those plates. So you can see in the electronic record, you have the plate IDs, you have the name of the user, and you have the particle count results all in one electronic record. So finally, let's take a look at the requirements for particulate testing in parenteral drug solutions and see how these tests themselves can be made more robust. Now, the global pharmacopoeias are harmonized in the fact that they all recommend a primary method for subvisible particulate counting in parenteral to be light obscuration, or sometimes termed light blocking. And this is the preferred method, as I said, for subvisible particle testing. They go on to say that if it's not possible to use light obscuration for some reason, for instance, if the solution is too viscous or too dark, then you can use a secondary method, which is the optical microscope. However, there's good reason why light obscuration is the preferred method. And of course, the reason for that is that the contaminating particles we find in parenterals are not conveniently circular, which would allow easier and more reproducible counting and sizing. Instead, the counting particles using a microscope is very subjective, and there are several methods aimed at standardizing the sizing of irregular stained particles. But unfortunately, there's not just one method. And the methods available include deciding which is the largest dimension and measuring that, or the alternative methods of Martin's diameter and Ferret's diameter. And the problem is they are not only subjective, but each method gives a different result. Now, light obscuration, on the other hand, uses a laser light to illuminate a measurement window. The intensity of the light is measured by a sensor and turned into an electrical signal. The counter is calibrated by passing various sizes of spherical beads through the counter, partially blocking the laser light, and the resultant change in the electrical signal can now be said to be equivalent to each particle's size. And now the sample under test is passed through and the contaminating particles will cause a reduction in the laser light reaching the sensor. They'll block it or obscure it. And the resultant change in the electrical signal is then compared to those of the calibrating particles, and an equivalent spherical diameter can be assigned to the particle. Now, this method is far more repeatable and it's non-subjective, and that's why they use light obscuration, or recommend that you should. So let's take a look at the compendial test methods for subvisible particle counting. We'll start with USP 788 and EP 2.9.19. The pharmacopoeias here are harmonized on the required compendial method, and of course it's not possible to test all the vials in a production batch because the vials have to be opened to be tested. So the pharmacopoeias ask that a minimum sample from each production batch of 10 vials be pooled and analyzed. However, they go on to say that 4 samples of 5 ml each are taken from the pooled sample, asking that the first sample results are discarded to prevent carryover from the previous test, and that the subsequent 3 sample results are averaged. Now, to accomplish this test, the pharmacopoeias require that the total pooled volume of these vials must be at least 25 milliliters as a minimum. So in many cases, more than 10 vials are required to achieve this minimum pooled volume, depending on the dosage volume in each of those vials. As I mentioned, the pharmacopoeias are harmonized on this testing, and the pass-fail criteria for small volume parenterals of less than 100 ml dosage form is based on the maximum number of particles at 10 microns and larger and 25 microns and larger. For large volume parenteral solutions larger than 100 ml, the particle sizes are the same, 10 and 25 microns, but the difference is the number of particles are expressed as particles per container for the small volume parenterals and as counts per milliliter for the large volume parenterals. So the way the counts are expressed is different. So now let's take a look at the relatively new United States Chapter 787. Now this is designed specifically to provide a compendial method for testing subvisible particulate contamination in therapeutic proteins such as vaccines and any large molecule biologic drug. Now the pass-fail criteria for the 787 is the same as it is for 788. And the difference actually lies in the required compendial test method. Unlike 788, 787 allows the testing to be carried out on just one single vial and calls for a minimum sample volume of just 0.2 milliliters. Now, if you remember, they wanted 4 lots of 5 milliliters for 788, so this is quite radically different. Now, applying this test method using particle counters can prove challenging, as the target for the sampler is much smaller because there's not lots of vials pooled into a beaker. It's just a single vial. And it often has a small conical base, which means it won't stand upright, and you have to have it exactly in the right place so that the sampler will take the sampler. So most particle counters, of course, have a relatively large tare volume, certainly large when compared to 0.2 milliliters, or dead volume, it's sometimes called. And this is the volume that must be pulled into the counter before the sample reaches the sensor and can be counted. Now, the HIAC 9703 sampler can be optimized for this compendial test by using the optional small vial clamp, holds the vial upright and in exactly the right location for the sampling arm to take the sample. And we have a sampling probe with a very small tare volume. It's only got 0.091 milliliters tare volume. So we can optimize this test for biologics testing. Now, as I mentioned, although largely harmonized, the requirements for parenteral drug particulate testing do vary, and particularly from product to product. The volume of the sample to be analyzed and the format the results are reported varies from product to product, as I said in the table. So the sampling requirements for small volume therapeutic proteins, such as vaccines, is different, very different to that of a large volume parenteral, such as an intravenous drip bag. Now, results must be calculated and expressed in the correct format, e.g. for small volume parenterals, it's counts per container. For large parenterals like drip bags, it's counts per milliliter. So The way the results are expressed depends on the type of product you're testing. So particle counters just do a measurement and deliver particle counts per milliliter. That leaves the operator then to calculate the number of particles per container or per milliliter. And of course, that manual calculation and interpretation of the results can invite opportunities for human error to creep in. So the compendial testing for subphysical particles can present opportunities for human error, as I mentioned, and subsequent data integrity challenges. So this I'm showing on the screen here is a fictitious example, but it serves to show what can happen. These are two fictitious drug products in the same size enclosure. They're both in 5 ml vials. Product A has a patient dosage volume of just 1 milliliter. Now, product B, however, has a patient dosage volume of 1.5 milliliter. Now, running the test for both product types on a standard particle counter delivers the same concentration of particles at 25 microns of 500 particles per milliliter for both these. Now, is this a pass or a fail for these two products? Now, with product A, there's just 1 ml in the vial. So with 500 milliliters of 500 particles per ml, the container itself only contains 500 particles at 25 microns. And this is a pass when you compare it to the maximum allowed per container of 600 in the compendial test. However, product B has exactly the same number of particles per milliliter, but because the patient dose is 1.5 milliliter, this container now has 750 particles per container in it, which is a fail when compared to the compendial limit of 600 particles per container. So you can see, do the tests on two different products and you get one passes, the other one fails, the test results are exactly the same. So it can be very complicated and messy. So whilst you can use a general purpose liquid particle counter for testing particles in parenteral products, what we've done with the HIAC particle counters from Beckman Coulter is you've optimized them for this application due to the complexity in the testing and the opportunity for human error. So the various compendial tests are all built into the HIAC software, and the HIAC will automatically calculate a pass-fail result based on whichever compendial test the user is using. As QC teams tend to refer to their product by the brand name to describe the product, the optional procedure builder module for the HIAC software allows the user to name the compendial test by their product names. So you have a list of the product names instead of a list of the compendial test, and all the user now does is simply selects the required test for each sample by selecting the product by name from a drop-down menu. The counter then automatically applies the correct compendial test for each product, and there's no manual calculations as the correct compendial pass-fail criteria for each product are applied and a report is automatically generated. So I'm coming to the end of my presentation now. I'd like to summarize the key points. In our industry, it's very common that we typically focus on retraining when mistakes or data integrity issues happen. However, the underlying issue we're facing really is that retraining may not be the best solution. Manual SOPs are just not robust enough to prevent future recurrence of data errors and omissions. So QC instrumentation that automate the quality control SOP can potentially both reduce the impact of human error on the data integrity, but also bring welcome cost savings by reducing the time technicians need to devote to manually configuring the instruments and carrying out multiple pass-fail calculations. So thank you for your time today. I appreciate you joining me and listening. So this is the end of the presentation. So let's take a look at any questions that have been posted during the session.
Thank you, Tony, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. All right, let's go ahead and get started. So Tony, your first question says, thank you for your presentation. Please, can you tell me how the MET1 3400 ensures that the technician is using the correct version of the routine environmental monitoring SOP?
Great. Thank you very much for your question, and thanks for attending today. So the SOP version control is actually done inside the particle counter itself by the manager or the administrator. And once they've set it up and they're happy they've configured it in the counter according to their SOP correctly, they release the SOP for use on the counter using their electronic signature. And now the SOP appears on the environmental monitoring technician's screen ready for use. And also the SOP name and the version number that was used at the time of the routine environmental monitoring is actually included in the electronic records generated by the counter. So it's very easy to check which SOP was used.
Thank you. All right, your next question. How does the PAT700 ensure the integrity of the PDF electronic records that it generates?
Thank you. It's a good question. In fact, in the 21 CFR Part 11 guidance from the FDA, PDF is one of the recommended file types. And they suggest that because you can read PDF on many different platforms, Android, Microsoft, etc., etc. So the idea is you should be able to read that record well into the future. And the way we ensure the integrity of these PDFs is the PAT applies a randomly generated password to each PDF, which secures them and prevents them from being changed.
Okay, and let's go ahead and wrap up with this last question. How do I know if the technician used the correct compendial method on the HIAC 9703?
So, in fact, the compendial test method control, again, is done via the software in the counter. And the compendial test method and its version number are included in the electronic pass-fail report generated by the HIAC, just as we saw with the MET1 for routine environmental monitoring. Now, additionally, the technician chooses the appropriate test from a drop-down list, in this case, just using their own product names. So they don't even really need to know which compendial test to use with which product. They simply look up the product name in the drop-down list, select it, and the HIAC automatically applies the correct compendial test method according to their product.
Thank you, Tony. And do you have any final comments for our audience before we go today?
No, thank you. I just wanted to say thanks again for attending, and I hope it was informative and useful.
Well, thank you again, Tony, for your time today and your research. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. A reminder, questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.